And so this session includes two fantastic panels under the theme of reimagining what is possible. So I'd like to invite the first panel to come on up to the stage. Includes some of the all-stars from the Stone Living Lab. In fact, both of these panels do, so you're in for a real treat. Folks coming up on stage include Christo, Jarrett Byrnes, Kirk Bosma, Daniel Lopez, and Melba Torres Sosa. Yeah.
All right, let's take a second. Good afternoon, everybody. My name is Jarrett Byrnes. I'm one of the scientists working on the Living Seawalls project here in Boston Harbor. We are going to take you through a little journey about, well, basically from the beginning through implementation and sampling of the Living Seawalls project. Some of you will be coming with us on the field trip tomorrow. You'll see a little bit about what we're talking about, but we're really going to go soup to nuts, start to finish. Each of us should talk about 5 to 7 minutes, and then that will leave plenty of time for you all to ask us questions and open the floor for discussion about implementation of nature-based approaches with whatever project you're working on. So we're going to talk about this with respect to our Living Seawalls project, but I really hope that the general principles of what we lay out here you will find useful for whatever NBA that you are interested in thinking about. So we'll lead off with Joe.
Thanks, Jarrett. Is this on? Oh, there we go. Thank you, Jarrett. Hey, everyone. Joe Christo, co-director of the Stone Living Lab and Chief Resilience Officer at Boston Harbor Now. I'm going to walk through a couple of slides here that kind of set some context for this project. I think I can advance them here. Yeah, exactly. So as folks know, some of these slides may look familiar, but as folks know, the lab is just this really special and unique partnership that we're so thankful to have you all joining with us today and sort of being able to celebrate that and celebrate all, our work together for everybody in this room. But really these core partners are just so essential to the work we're doing, from Boston Harbor Now to UMass Boston to National Park Service, Department of Conservation and Recreation, the Executive Office of Energy and Environmental Affairs, the City of Boston, and our partnership with members of the Massachusetts tribe as well. The mission and vision of the lab, I think, has been pretty present throughout this conference, but just wanted to have that up on screen for folks as a bit of a reminder. And then talk about, the different areas of work that we're doing, right? It's the research and monitoring work, the education and engagement, our policy innovation work, and the climate preparedness work, of course, too. The lab really first started to come together as an idea in 2018. As Kathy Abbott talked about this morning, following those two storms in January and March of 2018, and I thought Kathy did a great job of covering that. And it really formally launched in 2020. We were a COVID baby, and then in 2021 through 2022, we really started to develop our programs and our research projects. And then that continued to refine that in 2023 and 2024, which included, the first conference of the lab and really starting to hone in on the 5 core research projects, one of which we're going to go into detail on this panel, obviously, the local context of Living Seawalls. And that's when we get to 2024 and 2025, actually launching that Living Seawalls project. In addition to the Living Seawalls project, we have our Cobble Berms project that people on the stage and in this room were so instrumental in as well. We have our real-time monitoring of Boston Harbor project and many others too. Education and engagement work is kind of core to what the lab's doing too, and it's so connected to the research, right? They go hand in hand. The Cobble Berms project really, I think, highlighted that in some really key ways on the field courses that Kirk and Brittany Hofnagel and Rebecca Shore and so many others were leading at various Cobble Berms sites. And of course, we do the same with the real-time monitoring project, with Rebecca leading our Wicked High Tides project out here in collaboration with the Boston Harbor Now team. just going through some nice pictures there of some of the programming. And our Teacher Institute, too. Don't we have some Teacher Institute alumni in here? Are the Teacher... They were before. Maybe they popped into another room. But just love the Teacher Institute, too. Just really core to what the lab's doing also. So, how did we get to the Living Seawalls project? That's sort of the context that brought us there. But in 2022, when the Earthshot Prize was coming to Boston, we got a call from the Earthshot organizers and said they have this amazing finalist from the 2021 prize called Living Seawalls, and they think it could be a really good fit in Boston. And we very shortly thereafter got on a call with Katie, quickly realized, wow, there's amazing synergy there between what Living Seawalls and the lab is doing, and really started to put together some ideas. Even before the Earthshot Prize happened, but then it did, and we were fortunate to meet the Prince and Princess when they were here visiting for it. You know, this was taken on the single coldest day that I'd ever experienced on Boston Harbor. I think it was crazy winds. It was it was amazing. But that was in December of 2022, right, Katie? Is that yeah? And from there, we went through, about a year and a half of, project ideation, site selection, permitting, making sure the funding was assembled, and then really starting to think through, fabrication and installation, and having just such great partners, locally here with Woods Hole Group, and then eventually with Sumco on the installation, and of course with Katie and her team in Sydney as well. And the two sites we landed on, which Kirk is going to go into more detail on, but we're one in the seaport, right at Fan Pier, and then a very different site at Condor Street Urban Wild in East Boston. So the seaport, I'm sure most folks in the room are familiar with that project site, but that is a very densely developed sort of urban neighborhood that is really a lot of newer buildings. These are former parking lots in that neighborhood. The Condor Street, and it's a little more exposed to the open ocean. It's still harbor, but it's a little more exposed. The Condor Street Urban Wild site is fascinating too, because it's right on Chelsea Creek. It's in an environmental justice neighborhood. It's a former brownfield site. It's a working waterfront, and there's amazing marine life there as well, which you're going to learn more about during this conversation here. So it's two really distinct but very complementary sites, and that was by design. So Kirk's going to go into a lot more of these details, but once we got to installation beginning this fall, that was when we were really partnering with the Sumco team. Thank you, Melba, for designing that amazing sign that was at the site in the seaport. And then, we jumping ahead here because we're going to fill in some of the details. Had some really nice press coverage highlighting the project as well. And that's what the finished project is looking like now. And the team celebrating it there back in November at our media tour, and habitat housewarming, we called it. So that was kind of the broad overview of how we got from having the idea of a lab in 2018 to launching this project in 2022 to actually installing it in 2024. I said I'd cover that in 5 or 6 minutes. I hope I did so. But I want to kick it over to my colleagues here to now go into much more detail on this project. So thank you.
Thanks, Joe. Hi, I'm Kirk Bosma. I'm a coastal engineer, work with Woods Hole Group, but also engineering director at the Stone Living Lab. I'll talk a little bit about once we started thinking about this and kind of picked the sites. And I will say the sites were centered in and around Boston for very specific reasons. And as I go through this, you'll learn that they're pretty quiescent sites. So one of the big research questions we'll have is, well, what will happen if we try them somewhere else? There is a site going up in Gloucester soon that will have a little bit more wave energy, but I'll cover a couple other things like that. First of all, I'll say is permitting was, I thought, going to be really straightforward, but just to give a sense of how, and this isn't regulatory fault or regulations fault, but like because of where we were putting it in the fan pier, we had to spend about $1,000 in just notification of abutters of the project because there were high-rise apartment complexes right next door. So the permitting turned out to be a little bit more than we originally expected, but at the end of the day, everybody was excited to get these in. In terms of the engineering and construction challenges, there were definitely some here because this was a new location for these really awesome looking tiles that we're gonna put in. And some of those specifically was the really large tide range we have here. I think Katie mentioned the other day, 2 meters, and I'll try to stay in metric units here the best I can. 2 meters, in Australia, but here we're really looking at 3.5 meters of tide range. And that upper picture there, you can just see that's at the fan pier site at low tide. And then if you look at the actual seawall that's there, that dark mark in there, you can see just how high the water can get, right? So it's a challenge from just laying these out, but really in terms of construction, it became a really big challenge. Seawall irregularities, which I'll spend a little bit of time on today, they're kind of like the roads here in Massachusetts, right? Like, I can't find my way around here. It's not like where I'm from in the Midwest, where everything's a square and very nicely laid out, and you can't really get lost. I don't know where I'm going half the time in roads. In the seawalls, you think they look really nice, but when we get into it, it's like they're not really just flat at any level, and so that was a challenge. And then something new for these tiles was certainly going to be the freeze-thaw cycle and ice impacts. Really, part of this test is not only the biological growth and ecology, which we'll get into in a second, but really the physical nature of these. How is the material going to really work under that? And that thaw cycle corresponds not just to the tiles themselves, right? If they have interstitial spacing where they're holding water for pools and stuff like that, what's going to happen there, but also between the tiles and the wall themselves. Are we going to crack those tiles right off of there during the ice impacts? So a big part of this monitoring is really related to that physical stuff too. And then wave forces is something we really want to get into. We talk a lot about, these first two sites are really about the ecology and what's going to grow on them, and we'll talk a lot about that. But we really need to look at how these things might affect physical processes too, and which I'll talk about again in a second. Just some pictures here. Again, this is at Condor Street Urban Wild. So if you look at it from far away, it looks like a pretty nice seawall, like pretty vertical straight, like some granite rocks stacked up. But if you really zoom in on it, there's all kinds of irregularities, right? There's gaps and holes and rocks sticking out at you and sticking back in there. And so part of the trickiness of this from the construction and engineering standpoint and figuring out how many, what kind of forces we were gonna experience, how many bolts do we need to attach these things properly, was like, well, are we actually really gonna be able to attach these in a way? And we worked very closely with Sumco. They did a great job on the construction front for this. And just to give you some examples of what that turned out to be. So there's a tile in the upper corner there where, the design and the calculations we did for wave forces, we wanted to have at least 2 bolts in each tile. There's 4 specific holes that can mount that in, and it wasn't easy because you would line these up, and we wanted to have them aligned properly with elevations and rows and stuff like that, but in some cases we would hit a gap, and then we couldn't really attach it very well. So I kept going out there, and we had to identify specific tiles, and we put marks on them and said, you gotta fix this one because this one is not attached well enough, and we really were trying to avoid that situation where we had 2 bolts like at the bottom and then nothing above that, because we thought that's a pure mechanism for cracking or failure of these particular tiles. So that was a little bit of a challenge, but they really did a pretty good job at the end of the day. Fan pier was a different story. They had, again, these irregular seawalls. It wasn't flat, like they had terraces actually in the units. And then they had a slope at the bottom. So it was a slope structure to a vertical structure. So the design on that was a little bit tricky, and then you can see we worked it out with Sumco to have extended connections to in between the different tile rows, because we really wanted to capture that full tidal range effect and all the tile rows throughout that. So that was a little bit of a challenge too, but we worked through that, and it's there, and it's functional, it's working. So again, these sites are really great in terms of looking at lower energy environment, what can we get to grow on them, those type of things. And I'm going to show you a quick, how do I get the video to go here? Advance, okay. This is just a real quick video of the time lapse of the construction process. That is loud music, but this is the fan pier site. And what I really want to point out here is they didn't work at high tide. So they had to really work and function at low tide. most of the time and you'll see the tide will start to come up and then we skipped a whole time period because they weren't out there during high tide periods, and we really had to center the construction around that tidal cycle, even more so here at the Fan Pier site, where you can see they had the boat there. They were using almost like scaffolding to work on some of it, and once we got above in the upper levels, it was a little bit easier, but it was really a remarkable process what they did to get those all ingrained and put in, and Jarrett will talk about the actual array configuration of some of that as well. So where are we going from here? I mentioned the physical processes. So the 2 things or 3 things that we really are going to focus on going forward, where we're talking to the city about this, is can we really use these to then reduce wave run-up and overtopping processes? So a little bit of engineering in here. That one factor there is a roughness coefficient. We know that roughness factors on structures can reduce run-up and overtopping by up to 25%. So that's not an insignificant amount. Actually, the most interesting part of that is like grasses and vegetation has the biggest impact. So, hey, double benefit if I can get frictional effects due to the layout of the tiles plus growth on it that reduces frictional effects. Maybe that is a great thing to do with existing revetments and seawalls where we can include that roughness factor. But will that really work with these living seawall tiles? And then what about beach scour out in front of the seawalls, right? So could we reduce that downforce of a breaking wave hitting the seawall? Everybody should be familiar with that seawall's induced scour seaward of the structure. Can we then reduce that in front of those seawalls? What I'm not promoting is that this is a check mark to like put seawalls up everywhere. That's not the point. This is a point where there are seawalls and they have to be there for some reason or barriers, can we green those up? Can we make them work a little bit more functionally in a nature-based perspective? And then will these actually work when we get high wave forces? Are we gonna like just destroy these things? So, we're looking at new locations. One area that really is intriguing to me is the Deer Island wastewater treatment facility, which has a nice smooth seawall in that area that we could attach these to. It does see a lot more wave energy, and then we're talking through, and by no means is this a design, I just found this picture on the internet of these tiles, but could we design tiles in a way that are more based towards roughness coefficients that reduce that run-up and overtopping process and or reduce that downforce that goes on the beach? So we're considering a couple different sites at a beach location that actually sees water on a pretty regular basis. Could we reduce the scour impact, compare that to a control site next door where we don't have the tiles, and then also somewhere where we know we're going to get that run-up and wave overtopping on a pretty regular basis? Can we reduce that volume of flood risk associated with it in addition to all the ecological values? Now we're going to get into the fun stuff. So.
Oh, great, thank you. Hi, so again, I'm Jarrett Byrnes. I'm an associate professor here at UMass Boston. Hope you're enjoying our campus today. I'm going to talk get into the weeds a little bit and talk about some of the, kind of the nerdy details about design. How do we know if living seawalls actually enhance nature? Actually very appropriate as I'm teaching my students experimental design this week. So how do we know if these are actually doing something? How do we know what is happening? As we began this process, we went through a site selection process in order to find ideal locations for testing some of these ideas. We wanted to know, first off, are our sites accessible to us? Can I and my students actually get down there and regularly sample them? Are they visible to the public? We wanted this to have an outreach component so that people could see them and see what is going on. We were looking for areas with a large enough seawall area that we could sample not only the seawalls but other areas around it. They needed to be vertical so we could have good comparisons to what's been done in the past, and minimal shading. So we were really interested in looking at the kind of growth you would see in the rocky intertidal instead of seeing the kinds of things you would see growing on piers and jetties, many of which are invasive. Lastly, we wanted a wall that actually was easy to attach to, although as Kirk can tell you, maybe we didn't think about that last one quite so much. In considering all of these pieces, and all of us in this room who are making decisions about when and where we should be installing nature-based approaches, we want to be very mindful of the fact that those decisions, which often aren't necessarily due to the biodiversity itself, may influence what we see. So the first question that we really had, so because of this we chose the Condor Street Urban Wild and Fan Pier after looking around at the harbor, consulting with Katie, and thinking about what might be ideal locations to install. And we wanted to know, first off, how can we actually look at these and understand whether or not any changes we see in biodiversity are due to the living seawall, or I don't know, maybe we installed them at an opportune time. We've had an incredible productivity boom actually this year in the Gulf of Maine for a variety of reasons. Maybe we're just seeing the results of that. This is a question we should all be asking ourselves. When we install an NBA, is it making a difference, or are we just seeing something spurious? And second, which panels actually matter? So we have a variety of species that live here in New England. What architectures will matter to who? And how does that inform how we design future living seawall installations? I don't know. I'll be honest, one of the reasons this whole process has been so exciting to me as a scientist is I have no idea what's going to happen. So there's some real fundamental discovery here. To answer the first question, we used actually something that's adapted from power plants, believe it or not. The San Onofre power plant installed in the 1980s was actually a hotbed of thinking about how do we assess impact. So from this we designed the before-after control impact. There's a huge literature in the 80s and 90s, lots of people slugging it out, arguing. It's quite fun, actually, if you're into that sort of thing. But the before-after control impact has really become a standard for environmental impact assessment. This idea that you're not going to maybe necessarily replicate putting in a power plant, so what do you do to know if it's actually done anything? You have to look both at the area where you're installing as well as other adjacent control areas, places that are very similar to it, and look at how they change over time, with the idea being that if you really are achieving an impact, you will see that that treatment site, that place where you're doing something, shift while nothing else does. And so while the patterns are still going to have a lot of fluctuations in them, there's going to be some sort of a shift. What's really interesting about this is in the environmental world, we've been doing backy designs for a little while. Economists have actually been running wild with these, using a slightly different name for it called difference in difference. And that literature has advanced a lot. So we wanted to actually use many of these developments in that world and do better than we typically do in before-after control impact, in environmental impact assessment. So because of that, here, for example, is Condor. We have 2 living seawall installation sites, but we actually have multiple controls per site, and they really differ. They are similar to the sites. You'll see that 2 of those controls are right next to the actual installations, but we have others as well, so we can get a good baseline of what is the environment, and how is it changing relative to what we're seeing in the seawalls. And what's actually interesting, Kirk mentioned that the permitting was a little bit more complicated than we anticipated. That actually was good for us because we could get 2 years of pre-sampling. So we actually have a much better baseline than just a one-off study. So that's impact. And again, I could talk about these kinds of designs for ages. Find me afterwards. I apologize for whatever I say. Second, what panels should we use? Yesterday, Katie presented this cornucopia of different panel designs that exist. And in thinking about what we wanted to install here in Boston, we're faced with a couple of questions. What matters? Now, these designs have some abstract designs, some that are more biologically based. And so we began by just saying, look, we want panels that are fairly generic in their features. So we want to control, because again, you always want something bare to compare to know, are these panels and their architectures really making a difference? And then crevices and rock pools seemed like a very logical place to start. Those are common features that we have here in our rocky intertidal. But other things creep in, and I think that's part of the fun. We wanted to ask other questions, like does size matter for rock pools? What if we had small versus large rock pools? And lastly, there were some tremendous results looking at algal growth using the swim-throughs, which I find funny. The swim-throughs were made for fish to swim through, and yet they seem to be fantastic for algae. And I'm all about algae, as anyone who knows me can tell you. And so this was another panel that we're very excited about, and something that the Living Seawall group was really excited about us installing here, particularly to see how things compared against the installation in Plymouth and soon Cork. Okay, so we have 5 panels, but types, but we only have a limited amount of space to work that in, and that leads to other design challenges. When we're installing any sort of NBA and we want to look at different variation, we have to think about this from the perspective of designing a good experiment, right? We know that panel effects could change at different tide heights. We're often installing these installations at different tide levels. We want replication. You want to know, a one-off isn't going to give you the information you need about what impact you have. And also funny things, right? If you really want to know what these panels are doing, no 2 panels can touch. Otherwise, you get all sorts of different strange outcomes that are not what you're actually testing. Classically, this would be really easy. We have something called the randomized controlled block design, which sounds very fancy, but it's not. It's just kind of arranging things in different patterns so that you have every row and column has at least one of each treatment, etc. But squares are not hexagons, and this was a real interesting challenge. And so we went through, and this led to a lot of, I think, really fun design and algorithms. And you too can now, if you'd like, design your own living seawall. I have the URL up there. Sorry, it doesn't have a QR code, and check it out. So again, as you're designing your NBAs, you have to think like an experimentalist and really think about what you want to do. All right. So now let's get to the fun stuff. So this is, again, the design still comes out, I think, looking fantastic. This is the Seaport seawall, and I just think it looks so interesting, even implementing all of these different principles. So I'll wrap up there, and just to remind you, anytime you're installing an NBA, it is an experiment. You have to think about it that way. It's not a one and done. You've got to monitor, you've got to look at it, you've got to think about design using best practices. And so for thinking about that monitoring, let's move on, and we're going to talk, I'm actually going to pass this over, Daniel, to Melba, to talk a bit about microbial communities, and then we'll move on to Daniel to talk about the macrofauna.
Hello. So I'm going to tell you a little bit about microbes. So behind the title here, we have some panels this was a week after installation, and you might look at them and think, well, they look cool, but there's not a lot going on. And you would be wrong, because there is a lot going on, because microbes are actually starting to colonize the surfaces. So in marine environments, microbes are among the first to land on surfaces, and they produce biofilms. And a biofilm is just a surface-attached community of microbes, and then they secrete these extracellular polymers that they use to protect themselves from environmental stressors. So here we have a photo of a biofilm, an electron microgram. I didn't do this. All of the biofilm photos I stole from someone's work. But to give you another idea of what they look like, the little circles in this photo are polystyrene, little balls of them, like little plastic balls, and you can see the biofilms forming around them. You cannot see the microbes in the photos with the yellow, but you can kind of see the microbes on the big photo where certain structures inside the microbes are highlighted. But why should we care about microbes? Well, it turns out that microbial metabolism is actually nature's most diverse toolkit. Any kind of metabolism that an organism can do, microbes can do. There's a microbe out there that can break down probably everything, even if we haven't found all of them yet. They're also important primary producers. They're important for nutrient cycling, and they are really important for pollutant degradation as well. For the catastrophe that was the Deepwater Horizon oil spill, their estimates range from 20 to 60% of the oil was actually degraded by microbes that were already in the environment. So we can really, if we want to understand how to clean up our oceans, we need to understand this microbial metabolism. Additionally, they're also really important for colonization of other species. So if we want to recruit specific macroorganisms, or I'm going to call them macros, to the panels, you might be able to use the specific microbes that we know attract the macros we want. So my research questions, I have a couple. Start with, do specific panels select for specific microbes? And we're talking about taxa here, so what kinds of microbes, what species of microbes? do specific panels select for specific metabolisms? So maybe the microbes look kind of similar between the panels, but they're doing very different things. So what genes are we recruiting for in specific panels? I'm interested in the relationship between the microbes and the macrobes. So what do we see with specific communities, and do these seem to be attracting specific macrobes that we want? And finally, if this relationship seems significant, can we actually seed panels before deployment with specific microbes that we can assure or at least enhance the success of these panels by recruiting species that we know we want on them? So to do this, and Danny is going to talk a little bit more about the sampling that we do out there, but for me, I use this very sophisticated Lego block to standardize the area. And I go out with sterile swabs, and I swab every panel. So we have one of our lab techs out here. We get a lot of help because we have a lot of panels, so we couldn't do any of this without our lab techs. And we take those samples back to the lab, where I freeze them until I can sequence them. And I'm still in the early stages of sequencing, but for any microbe nerds or genomics nerds out there, I'm doing 3 different types of sequencing. I'm doing 16S, which is going to select for a specific gene that is in all bacteria. I'm doing ITS sequencing, which is going to select for a specific region in all fungal genomes. So I'm looking at bacteria and fungi. And then I'm doing shotgun sequencing, which is just going to sample everything. But this is a little bit expensive, so we're going to only be doing this for select samples. But shotgun is going to give us more information about the gene pool that is in specific panels, as well as phages and viruses and other organisms like protists. To give you an idea of what this data looks like, on the right, I have a bunch of different samples. Each column is a sample, and you can see what bacteria taxa are making up each of the samples. And then before I hand the microphone to Danny, I wanted to show a little bit more biofilms, because I think microbes are really overlooked, and they can be very beautiful, and they don't only cause disease. So surfaces in the marine environment include live surfaces. So this is a kelp blade. In the first image at the top, you can see, so the blue in the bottom, that's the kelp, and on the top you can see the microbial community, which is actually a couple micrometers thick. You can see that there's just so much going on. They're really like little tiny microbial cities, and I'm really excited to be looking at them on these panels. Hopefully, if we get enough funding, I will be able to do some of this microscopy to also look at what the community physically looks like and what microbes are interacting with each other.
We're just going to switch presentations here. So hi, everyone. My name is Daniel Lopez, and I'm going to be talking a little bit more about who lives on the seawall, as well as the standardized international protocol that we're using to kind of monitor the biodiversity along these seawalls. So I'd like to start off giving a little bit of background about what a seawall habitat is like. First and foremost, you have to have a seawall there. It's pretty obvious. But these are generally characterized by a pretty simplistic substrate. They're really generally flat. They don't really have a lot of cracks and crevices, and lacking critical intertidal habitats that we would generally see, such as rock pool and those cracks and crevices that I described before. In addition to that, you're taking the expanse of a natural intertidal habitat, like we see over here. This is actually off of Pettix Island. And you're condensing that into just a few meters along a vertical gradient. And so as a result of what we're seeing here, the natural habitat and the species that would live there normally have a harder time adjusting to these seawalls due to the lack of variability and these critical microhabitats that can help support them. So what we generally find along these seawalls are more non-native species that can actually better adapt to these areas, or a lowered biodiversity overall within these systems. And so I was kind of brought on with this project as doing the background sampling as well prior to the installation and characterizing what the biodiversity looks like out here in Boston Harbor. We're using this international protocol that was brought on by the Living Seawalls project as a way not only to standardize our protocol for the installation of these seawalls, but also for the broader impact that the Living Seawalls have, having a global expanse. So a lot of this kind of comes from doing these quadrat surveys that I have out here. So that little PVC 25 by 25 centimeter quadrat, we measure along different heights along these seawalls, looking at the types of species that are growing directly upon the seawalls themselves. So what we call primary substrate, you can think of like a barnacle growing on the wall, and then secondary substrate or secondary cover, which is the species that are growing directly on the like the barnacle. So getting that two different layer interaction on those seawalls. Additionally, we're interested in monitoring the types of mobile species, so crabs and snails that are living in these areas, and we're just counting the number of those that we see within the boundaries of that of those quadrats. Additionally, we're really interested in not only the species that reside within the low, we can measure during the low tide, but we're also interested in seeing what kind of fish species that are living along here, and trying to understand the diversity of the fish that are along these seawalls as well as the behavior of them. So part of this part of adapting this protocol has been using a camera monitoring system where we put cameras out to look at the seawall during the high tide for about an hour to get an hour's worth of data, where we go back into the lab to annotate those videos. As we had mentioned before, some of the challenges that actually can come with working like with an international protocol are the differences in the sites themselves. So what Kirk had said, the intertidal here is significantly larger, the range. So instead of using a single camera maybe to look at these areas, we actually have to use 3 different cameras along different positions of the seawall, and we're dropping them over the seawall itself to kind of hang over and looking directly at these areas. I will say, if any of you are in the Boston area and you're out in the summer and you see these big PVC setups with concrete bricks on them, that's me. You're welcome to drop by and say hi. I love talking about these things. So I kind of want to talk a little bit more about what we saw. So kind of touching on the idea of the species that we see and the biodiversity. There's not a lot, but there is life there. And it's really interesting to see how these areas have kind of adapted to these substrates. So we have different algal communities that have managed to kind of live. It's more patchy than you would regularly see in a New England intertidal. Resident crabs that we have within the area are the Asian shore crab. We also get a huge amount of littorina snails that kind of occupy these areas. And even in those cracks and crevices, we do occasionally get mussels. Additionally, we get rats. Comes with the territory of working in an urban harbor system. And we've aptly named the boat that we use to do some of our surveys the algae rat. Taking a little bit further, some of the other stuff that we've seen are much larger species of crabs, despite the coloration that you see there. That is actually a green shore crab. We get really a non-native tunicate, the Botuloides violaceus, in the middle with the bright orange. And then we also have some of our native Ascophyllum nodosum within these areas. So some of the fish video that we've come across has been really interesting and fun to see in these areas. Actually, initially when Jarrett and I had talked about doing this site in particular at Condor, we were always talking about, well, it's a highly, heavily industry area. We didn't really expect to see a lot, but despite that, we still do see some. So these are mummichog killifish species that have been swimming around. We also have this very action-packed scene of crabs fighting to hold on to a blue mussel that this cunner has been trying to fight. So just, it's been a treat going through this video and seeing what we've come across. Really neat stuff. And so I kind of want to talk about what's next. So I've done this biodiversity sampling for the past 2 summers, getting an idea of what we're seeing. But now that the panels have been installed, going forward, we're going to take that sampling and continue that monitoring much more so than we've been doing prior. We're doing seasonal sampling to watch the progression of these panels colonize. And additionally, looking at specifically the inner and outer areas of microhabitats. From what I have seen so far doing some of the sampling, there's been a lot of interesting growth, some that I'm not familiar with, which makes me have to go back and freshen up on my species IDs going into the summer. But we're really excited to jumpstart into the summer sampling that we're going forward and seeing what kind of species colonize these panels. And thank you.
Here, there we go. Thank you, everyone. I think we have time for at least one question, hopefully. I know these folks will probably get a lot of questions later this evening. So let's see, any questions? Oh, yep.
Should you do a condition assessment of the seawall so that once these tiles are on, they're going to stay on there for 10 to 15 years with the wall behind it start damaging?
Yeah, we did an engineering assessment of the seawalls before attaching the living seawall piles for 2 different reasons. One, to make sure that the tile attachments wouldn't impact the actual structural stability of the tile, of the seawalls themselves. And then also we had to do the calculations just to make sure that the tiles themselves wouldn't pop off. What we don't know very well is what's going to happen with tiles with the ice impacts. That's something we're going to learn a little bit more about. There's also different materials you can make the tiles out of. That's something I think we're going to investigate as we go forward through time. And then the tile designs, which, Katie, you can correct me if I'm wrong on this, have been primarily geared towards ecological and biological species growth. I think we're thinking about changing some of those up a little bit to think about some of the physical parameters as well.
Great, thank you. Maybe one quick question here.
Hi, everyone. Great talk. First of all, love the team effort. And I'm wondering if you think of micro to macro as a form of succession on the seawall, and I'm wondering if the pace of academics and publishing are affecting the site selection criteria at all, and if timing was not a factor, and if so, if timing was not a limiting factor, if those criteria specifically, minimal shading is what I'm curious about, would change. Because, I guess I'm wondering if you're prioritizing photosynthetic activity at the beginning, where heat might be a limiting factor to macro diversity later on.
I can talk about that with respect to site selection. No, it really, we were trying to choose sites that that were comparable to work that's been done in the past. We're trying to sort of build on an amazing edifice that's already out there, but try and understand how that translates to a completely different biogeographic region. So in the future, we've already been talking about a wide variety of different types of places that they could be installed, and certainly the effects may be different, but we're starting where we're starting. And then with respect to succession, I'm actually, I'll be honest, we started the project without Melba, which really was unfortunate. And Melba came and joined my lab later, and it just seemed like a perfect fit to begin to understand that successional process. So that was, I think, very much in mind, and Melba was talking about the ideas of pre-seeding. These are all ideas to see, are there ways that we can use the natural history of New England, particularly developing a natural history of microbial systems, to take advantage of that successional process to get the kinds of biological communities that, that are flourishing and resilient.
Thank you so much, everyone. I think we'll wrap up that panel, and feel free to find these folks afterwards for some more questions.